For over a century, scientists thought they had the saber-toothed tiger figured out. Big cat, long fangs, extinct, done. Then DNA entered the room and everything changed. What researchers pulled from ancient bones, some of them over 10,000 years old, didn't match the story we'd been telling. The family tree was wrong, the behavior was wrong, and the reason it vanished? That might be the most wrong of all. Stay with me, because what the genome of this animal revealed has quietly rewritten one of the biggest mysteries in prehistoric science. To understand what was lost, you first have to understand what the saber-tooth actually was, because the name misleads almost everyone who hears it. Smilodon was not a tiger. It was not even closely related to tigers. The last common ancestor between Smilodon and any living cat existed roughly 20 million years ago, so far back that modern lions, leopards, and jaguars are all more closely related to your house cat than any of them are to the saber-tooth. Smilodon belongs to a completely separate branch of the phalid family tree called the Machairodontinae, a lineage that produced multiple independent saber-tooth forms across tens of millions of years, on multiple continents in multiple ecological roles. The canine teeth were not a trick, not an accident. They were a solution that evolution arrived at again and again, independently, because it worked. The saber-toothed cat was not a tiger that grew strange teeth. It was a completely different experiment in what a large predator could be. Smilodon fatalis, the North American species most people picture, weighed between 160 and 280 kilograms. That puts it in the weight class of a modern lion. But the resemblance ends there. It had a shorter tail, a more muscular forelimb structure than almost any living cat, and a jaw that could open to an angle of approximately 120 degrees. A lion's jaw opens to roughly 65. Those canines, sometimes exceeding 28 centimeters in length, were not used to crunch through bone. The teeth were too brittle for that. Current biomechanical modeling suggests Smilodon used them for a precise, targeted kill strike, most likely into the throat or underbelly of already restrained prey. This was not a blunt instrument. This was a scalpel. And it hunted successfully on this continent for over 1 million years, from the early Pleistocene through to its extinction roughly 10,000 years ago. By the time Smilodon finally vanished, it had already outlasted dozens of other species, survived multiple climate fluctuations, and adapted to ecosystems from Patagonia to what is now Los Angeles. The La Brea tar pits in California have yielded over 166,000 Smilodon bones. One site, one city block in Los Angeles. 166,000 bones. This was not a struggling species on the margins. This was a dominant predator at the height of a lineage. When a predator as successful as Smilodon disappears this completely and this quickly, people reach for dramatic explanations. The most persistent of these is the human hunting hypothesis, the idea that early Paleo-Indian peoples who entered the Americas via the Bering Land Bridge simply hunted saber-tooths and their prey species to extinction within a few thousand years. The overkill hypothesis, as it's formally called, was championed most forcefully by geoscientist Paul Martin, beginning in the 1960s, and has never fully left the literature. It has vocal supporters. It is also, when examined closely, incomplete at best, not because humans couldn't have contributed to the extinction, but because the hypothesis in its strong form attributes a level of direct persecution to Smilodon specifically that the archaeological record does not support. We have mammoth kill sites. We have horse kill sites. We have no confirmed Smilodon kill sites. No saber-tooth bones with embedded projectile points. No butchering cut marks on Smilodon remains. If humans hunted the saber-tooth directly, they left no physical trace of it. The second popular explanation is pure climate determinism. The Pleistocene ended, the megafauna the saber-tooth depended upon collapsed first, and Smilodon starved into extinction as an ecological cascade. The prey went. The predator followed. Plausible. Tidy. And yet the climate had shifted dramatically before, multiple times, across the full 2 million years of the Pleistocene, and Smilodon had survived every prior oscillation. The Younger Dryas cooling event, around 12,900 years ago, was severe, but it was not the first severe climate event these animals had weathered. Climate change alone does not explain why this transition was the one that finished them when the previous ones had not. A third fringe narrative, popular in certain online communities and occasionally in popular science writing, frames the saber-tooth as an evolutionary dead end. The argument goes, those teeth became too large, too fragile, too metabolically expensive. Smilodon overspecialized itself into an evolutionary corner, and extinction was simply the inevitable terminus of its own excess. This interpretation feels intuitive. It satisfies a narrative of hubris. It is also not supported by the fossil record. The Machairodontini was producing saber-tooth forms successfully for 30 million years, across dozens of species, across multiple continents. The trait persisted because it conferred advantage. You do not sustain a 30 million year dynasty with a fundamentally bad design. The teeth that look like excess to modern eyes were precisely calibrated killing tools that worked reliably for geological epochs. A fourth theory, less common but occasionally surfacing, suggests disease, a novel pathogen entering the Americas either with human migrants or through ecological disruption could theoretically have swept through megafauna populations. The problem is evidence. Ancient DNA can, in principle, detect pathogen signatures in host tissue, and current Smilodon specimens do not show the patterns consistent with a catastrophic disease event. This doesn't eliminate disease as a contributing factor, but it removes it from the center of the explanation. The hidden assumption beneath all four of these theories is the same one. They all treat Smilodon's extinction as something that happened to the animal, an external force striking a passive population. Climate changed, humans arrived, prey vanished, pathogens swept through, and Smilodon simply could not respond. What the ancient DNA evidence reveals is that the story was far more complicated. Smilodon was not passive. Its genome shows a population that was, right up to the end, genetically healthy enough to adapt. What it encountered was not a single pressure point. It was a trap, assembled slowly from multiple directions in a way that its 30 million years of successful strategy had never prepared it to escape. For most of the 20th century, everything we knew about Smilodon came from bones and teeth, morphology, structure. The architecture of a predator reconstructed from its skeleton, the way you might reconstruct a cathedral from its foundation stones. Impressive, informative, but fundamentally silent about the living thing that once occupied it. Bones tell you shape. DNA tells you history. That changed meaningfully in 2024, when a team of researchers published the first successful ancient DNA extraction from Smilodon specimens, a technical achievement years in the making. The challenge is preservation. Ancient DNA degrades. It fragments into shorter and shorter pieces over thousands of years, contaminated by bacteria, fungi, and the DNA of every organism that has touched the specimen since death. Extracting readable genetic material from a 13,000-year-old predatory cat buried in an asphalt seep in California is not a routine laboratory procedure. It required advances in library preparation, the method by which fragmented ancient DNA is assembled into something a sequencer can read, and computational methods capable of distinguishing genuine Smilodon sequences from the avalanche of contaminating DNA surrounding them. The fact that it succeeded at all is one of the more quietly significant moments in recent paleogenomics. What they recovered was partial. Ancient DNA almost always is. The completeness of ancient genomes exists on a spectrum. A mammoth frozen in Siberian permafrost yields far more than a cat buried in California asphalt at a much warmer mean temperature. But partial does not mean uninformative. Even fragmentary genomic data, when analyzed correctly, reveals population structure, evolutionary relationships, and the genetic health of a lineage in ways that no amount of skeletal analysis can replicate. The first layer of the picture, evolutionary distance. Smilodon's genetic divergence from living felids was confirmed to be even deeper than the fossil record had suggested. The molecular clock, the rate at which neutral genetic mutations accumulate in a lineage over time, pointed to a separation so ancient that Smilodon was genetically almost a different kind of cat entirely. Not a tiger, not a lion, not even a relative of either in any meaningful recent sense. Something older, developing along a completely independent evolutionary trajectory for longer than modern Homo sapiens has existed as a species. Our entire genus, Homo, is roughly 2.5 million years old. The split between Smilodon's ancestors and those of living cats predates that by roughly 18 million years. This has implications beyond taxonomy. It means that whatever behavioral, physiological, and ecological adaptations Smilodon carried, its hunting strategies, its social structure, its sensory apparatus, were products of a lineage that had been optimizing independently for nearly 20 million years. We cannot simply assume it behaved like a large living cat, because it was not a large living cat. It was something genuinely different, running on a different set of evolutionary solutions. The second layer, population genetics. By analyzing genetic diversity across multiple specimens, comparing the variation within the Smilodon genome across different individuals from different time periods, researchers could begin to reconstruct what the population looked like in its final millennia. And here the picture becomes complicated in a way that upends the standard extinction narrative. The evidence suggests that Smilodon fatalis maintained relatively stable genetic diversity into the late Pleistocene, right up close to its extinction window. This is not what a species in long-term decline looks like genetically. A population that has been slowly shrinking for thousands of years accumulates a characteristic signature. Heterozygosity drops. Deleterious recessive mutations pile up because there are fewer unrelated individuals to mask them. The genome shows the constriction of a bottleneck. This is what we see in species like the cheetah, an animal that passed through a severe population bottleneck roughly 10,000 to 12,000 years ago and carries the genetic scars of it to this day. Cheetahs are so genetically similar to one another that skin grafts between unrelated individuals are rarely rejected. Smilodon does not show this pattern, not in the way a slow millennial decline would predict. The population did not slowly wither. It was not bleeding out over centuries. Something disrupted it rapidly, a fast-moving convergence of pressures that the genome did not have time to reflect before the lineage ended. The third layer, diet and prey dependence. The DNA evidence does not exist in isolation. It has to be read alongside isotopic data, and that combination reveals something critical about Smilodon's vulnerability. Stable isotope analysis of bone collagen measures the ratio of carbon and nitrogen isotopes incorporated into an animal's skeleton over its lifetime. Different prey species leave different isotopic signatures in the bones of their predators. Applied systematically across Smilodon specimens from different time periods and geographic locations, this analysis has refined our picture of Smilodon's diet with a precision that fossil teeth alone could never provide. The results are consistent. Smilodon was not a generalist. Across its range, across different time periods, it targeted large, slow megafauna, horses, bison, ground sloths, camels, and juvenile or weakened mammoths where available. These were not small or nimble prey. These were animals that required cooperative hunting, ambush, and restraint. Prey whose mass and defensive capacity meant that a solo kill was either impossible or catastrophically risky. The isotopic data suggests Smilodon may have been something closer to a specialist ambush predator, patient, precise, dependent on prey that was large enough to be worth the risk of engagement, but not so unpredictable as to be unhuntable. When those prey populations began to fracture under simultaneous climate pressure and human hunting activity, Smilodon's food base did not thin gradually. It collapsed in concert. The horses went, the camels went, the ground sloths contracted their range, the mammoth populations fragmented, and each disappearance removed another option from an animal that had not evolved to have many options to begin with. The fourth layer, comparative genomics and the survivors. Perhaps the most telling evidence comes not from Smilodon's genome alone, but from comparing it to the genomes of large felids that survived the same extinction event. The cougar, puma concolor, went through its own severe genetic bottleneck at the end of the Pleistocene. Genetic analysis suggests the modern cougar descends from a very small founding population that survived the megafauna collapse, likely by pivoting aggressively to smaller prey. Today's cougar hunts deer, peccaries, raccoons, rabbits, whatever is available. Its jaw mechanics, its limb proportions, its body size all sit within a range that allows this flexibility. It is not optimized for any single prey type. It is optimized for adaptability. The jaguar shows a similar pattern, a population bottleneck in the late Pleistocene, followed by recovery as prey communities reorganized around smaller species. Smilodon has no modern survivors to compare, but the contrast with the cats that did survive tells the story clearly. The survivors were generalists. They bent. Smilodon was a specialist, and specialists, when their ecological niche disappears, face a choice between rapid adaptation and extinction. Rapid adaptation requires either genetic diversity sufficient to produce new variants quickly, or behavioral flexibility sufficient to bridge the gap while those variants emerge. Smilodon had maintained genetic diversity. What it likely lacked was the behavioral and morphological flexibility to redirect that diversity into a new survival strategy fast enough. The scalpel, when the patient disappeared, could not become a hammer. The fifth layer, what the genome cannot tell us, and why that matters. There is a limit to what ancient DNA can recover, and intellectual honesty requires naming it. We cannot currently extract Smilodon behavioral data from its genome. We cannot sequence its neural architecture, reconstruct its social complexity, or determine definitively whether it was solitary or cooperative. There is skeletal evidence, healed injuries on some Smilodon specimens that suggest other individuals may have provided food to wounded animals, implying some level of social tolerance or even cooperative behavior. But the DNA evidence is silent on this. Future advances in ancient epigenomics, the study of which genes were expressed, not just which genes existed, may eventually change this. For now, the behavioral picture remains partially inferential. What we can say with confidence is this: the genetic data rules out a slow decline. It rules out the evolutionary dead-end narrative, and it points strongly toward a population that was healthy until it wasn't, hit by something fast, something multi-directional, something that its 30 million years of successful strategy had never prepared it to survive. There's a habit of thought, comfortable, persistent, and wrong, that assumes the further back you go in human history, the simpler and less capable the people were. It saturates the fringe theories around ancient monuments. It surfaces in casual assumptions about Neanderthal intelligence. It quietly underlies the instinct to dismiss Paleo-Indian hunters as a serious ecological force, as though people without metal tools or written language must have been too primitive to reshape an ecosystem. The Paleo-Indians who entered the Americas were not fumbling survivors stumbling through an unknown wilderness. They were the inheritors of hundreds of thousands of years of accumulated hunting knowledge, ecological intelligence, and sophisticated stone tool technology, the product of a cognitive lineage that had been solving complex environmental problems since before Homo sapiens was anatomically modern. Consider the physical achievement of their arrival alone. These people crossed, or coastally navigated around, one of the most inhospitable regions on Earth to enter a continent they had no prior knowledge of. They had no maps, no compass, no written record of what lay beyond the horizon. The Bering Land Bridge crossing required sustained movement across tundra in glacial conditions, hunting as they went, reading landscape and weather through observation alone. The coastal migration hypothesis, increasingly supported by archaeological finds along the Pacific coast, proposes that some groups traveled by watercraft, island hopping down the coast of North America. Watercraft capable of open water travel, built without metal tools, navigated without instruments, in waters that could kill an unprotected human in minutes. And once inside the continent, they did not stop. Within a geologically brief period, estimates range from a few hundred to a few thousand years, their descendants had reached the southern tip of South America, Tierra del Fuego, 11,000 miles from the Bering entry point. Across every climate zone the Americas contain, through jungles, deserts, mountain ranges, and river systems none of their ancestors had ever seen. This was not accidental drift. This was directed systematic exploration of an unknown world, executed across generations, without a single tool we would recognize as modern. Their stone technology repays close attention, because it is easy to read stone tools as a synonym for primitive, and that reading is wrong. The Clovis point, the earliest widespread projectile point type in North America, dating to roughly 13,000 years ago, is a precision artifact. It is bifacially worked, meaning flaked on both faces to produce a symmetrical cutting edge. It is thinned at the base for hafting, attachment to a wooden or bone shaft, using controlled pressure flaking that removes thin, precise chips without fracturing the tool. To produce a Clovis point requires understanding the fracture mechanics of flint or chert at a level that took modern experimenters years to develop after dedicated study. The Folsom point, which followed Clovis technology and appears in the archaeological record around 12,000 years ago, is even more demanding. A Folsom point is fluted, meaning a channel flake is removed from the base of both faces, running upward along the midline of the point. This is done with a single, precisely directed blow of a specific tool at a specific angle, with specific force. If the strike is even slightly off, the point shatters. Accomplished Folsom knappers today, people who have studied and practiced the technique for years, describe it as one of the most technically demanding tasks in lithic technology. The hunters who produced Folsom points did so routinely in the field, in cold weather, after a day of hunting, with no second chances on a spoiled point. They understood the mechanical properties of stone the way a modern machinist understands metal, not through formal instruction, through observation, practice, and the accumulated teaching of generations. And then, there is the direct evidence of what they hunted. At Murray Springs in Arizona, excavated in the 1960s and 1970s, archaeologists found the skeletal remains of a mammoth, Mammuthus columbi, the Columbian mammoth, which stood up to 4 meters at the shoulder and weighed up to 10 tons. Surrounded by Clovis points and butchering tools, the kill site preserved evidence of systematic butchering, specific bones disarticulated in a sequence that reflects knowledge of anatomy, hides removed in a way that preserves maximum usability, marrow bones cracked for their fat content. This was not a scavenged carcass. This was a planned, executed, efficiently processed kill. At Blackwater Draw in New Mexico, one of the original Clovis sites, the remains of multiple mammoths and Pleistocene horses appear in direct association with Clovis points across a sequence of occupation layers. At Hebier and Schaefer, mammoth sites in Wisconsin, mammoth remains with butchering marks have been dated to roughly 14,800 years ago, potentially pre-Clovis, pushing the human megafauna interaction timeline even earlier. You do not take down a 10-ton animal by accident. You do not take it down alone or without coordination or without prior knowledge of its behavior, its defensive responses, its vulnerable points. Coordinated mammoth hunting required communication sophisticated enough to plan an ambush, assign roles, and execute them under pressure. It required knowledge of the terrain specific enough to choose a location where the animal could be driven or cornered. It required the manufacture and maintenance of tools capable of penetrating hide several centimeters thick. The assumption that early humans were too primitive to have significantly impacted megafauna populations was never grounded in evidence about what those humans were actually capable of. It was grounded in a failure of imagination, an unwillingness to look carefully at what the archaeological record actually shows and take it seriously. Here is what the DNA actually tells us, the complete picture, assembled from genetics, isotopic chemistry, comparative anatomy, and archaeology. Smilodon fatalis was not a failed experiment. It was a supremely successful one, a precision predator that dominated the Americas for over 1 million years, that maintained genetic health into its final centuries, that had survived every climate oscillation the Pleistocene had imposed on it before the last one. Its design was not flawed. Its 30-million-year dynasty was not luck. Every adaptation it carried, those canines, that four-limb musculature, that ambush-optimized body plan, had been tested and confirmed across geological epochs. What ended it was not a single cause. Not climate, not disease, not over-specialization, not a single dramatic human hunting campaign. What ended it was a convergence. The kind of convergence that a 30-million-year-old lineage, perfectly adapted to a world of predictable megafauna and stable ecological niches, had simply never encountered before. The climate shifted, contracting and fragmenting the grassland ecosystems that supported the large-bodied prey Smilodon required. Simultaneously, human hunters, flexible, cognitively sophisticated, armed with precision projectile technology, entered the system and began exerting pressure on those same prey populations from a completely novel direction. The prey could not sustain pressure from both sides, and as the prey went, Smilodon's options went with them, one by one, faster than a large-bodied specialized predator with a multi-year reproductive cycle could adapt to. The trap closed slowly, from multiple directions, and then all at once. The saber-tooth didn't lose to a stronger animal.